Welcome to the spinal cord dissection portion of this lab. So to begin, I hope everyone has a spinal cord with them. If you don't, just follow along and use the images in your slide to sort of follow along with what I'm explaining and saying and showing. So if you do have a spinal cord, get it ready. What you will also need is the scalpel as well as some forceps. Help you out. Now to begin with, what we're going to do is we're just going to take our intact spinal cord and we're just going to take a look at first the spinal nerves. So remember, those are what's exiting the spinal cord itself. So also, we'll just explain the spinal nerves first of all. It's these just protrusions coming out of the spinal cord, right? So sort of just yellow or anything that's sort of outside the spinal cord is the spinal nerve. But if you take a closer look, and if you have a good one, if you have a good spinal cord, what you can actually see is how it attaches to the spinal cord itself. You can sort of discern dorsal versus ventral rootlets. And in particular, what you want to really look for is the dorsal root ganglia. Now, it'll be difficult to see, but I do see a few on my spinal cord. And what will sort of help you out is, which is what we'll do in the next slide, is figuring out exactly what is the dorsal versus the ventral portion of the spinal cord itself, and that way you can tell, oh, on the dorsal side I'm going to most likely be seeing some dorsal root ganglia. So if you take a look at your spinal nerve and you see some sort of ganglia looking figure, then you might as well conclude that, hey, that pretty much might be the dorsal root ganglia. Just as a refresher, the dorsal root ganglia is what carries the cell bodies of the sensory afferent fibers coming in from the periphery to the spinal cord. So moving on, we want to identify the top and the bottom. So more importantly, the posterior or the dorsal section of the spinal cord versus the anterior or ventral section. So what you can do is, if you have a spinal cord and if you look at the two ends and they're sort of difficult to see, gray versus white matter, sort of like what's shown in your slide, you could just take your scalpel and just cut a new plane into the spinal cord, and then that hopefully gives you a better view. So looking at your spinal cord, I can see it in mine already. You should sort of see this X shape. Remember, that's going to be your gray matter. And the parts or the legs of that X that are sort of longer and you can see are connecting to the very surrounding of the spinal cord itself, those are what's going to be your dorsal gray matter. And those that sort of seem to be shorter legs of that X that's being made, those are your anterior portions of your gray matter. So with anterior versus posterior now figured out, you can actually go ahead and look for, they're very small, but the posterior median sulcus as well as the anterior median fissure. So again, they're very difficult to see, but there is small nicks in the posterior median section of your spinal cord, as well as in the anterior median section. So the anterior median is called a fissure because it's much deeper groove, and the posterior section, the posterior median, sulcus. So we call it a sulcus. Next, what I want you to do is actually take your spinal cord and take your scalpel and cut straight through, and be very careful not to cut all the way through your spinal cord, just enough to remove this outer layer, which is called the dura. So I cut through mine. And remember, dura mater means tough mother. So it's really that tough tissue that's sort of surrounding the spinal cord there. Now, with that done, what you can do is go ahead and separate it all out. And what you'll begin to see are these pretty similar, stretchy, sort of flexible, noodle-like figures. So the differences you need to make here is identifying a denticulate ligament versus a dorsal rootlet, or any rootlet in this case. How you can tell the difference is these rootlets, they sort of look more like noodles. And the denticulate ligaments are more sort of the stretchy connective tissue, right? More web-like in appearance. So the denticulate ligaments actually, they're pia matter. They actually protrude through the arachnoid and attach themselves to the dura, sort of to create this connection between the pia, the spinal cord, and the dura mater. So it's just a connector. And the dorsal rootlets, of course, they carry dorsal sensory fibers into the spinal cord. So again, the distinction, which you can actually see in your slide, these rootlets look more like noodles, whereas the denticulate ligament, which is again pia mater, that of course looks more like a connective tissue. Now, moving on, what you can do is actually go ahead and start to remove the the dura mater. And what you'll see, and some of you may have this stuck onto your dura if you cut it a little too deep, it's the arachnoid mater. So the arachnoid, again, it's sort of web-like in appearance, right? Hence arachnoid. And you can start peeling that off as well. And once you do that, right, you're going to see more clearly these blood vessels that begin to appear. And that's because, if you remember, these blood vessels reside between the arachnoid mater and the pia mater. So all that should be left is the pia mater covering the spinal cord. So last but not least, if you go ahead and actually just peel the pia mater off of your spinal cord, you can see those blood vessels actually start to tear right off as well. And you have a bare spinal cord here. Now again, with all of that pia mater taken off, it may be easier for you to be able to actually see the fissure, because the pia is what sort of encloses the fissure together, which makes it difficult to view, which made it difficult to view initially. So now hopefully you see a more distinct fissure, right, cutting through the center. And it'll still be difficult, but hopefully you can make out the sulcus on the posterior median aspect as well. So again, we're identifying the posterior median sulcus as well as the anterior median fissure. And with that, you have completed your spinal cord dissection. The following slides are going to give a little more in-depth review of the different things we just talked about. And of course, if you have questions, do bring them up in office hours. Thank you. So taking a look at the model, and you can of course follow on slide 11, we have two distinct regions, one being the gray matter region and also the white matter region. So gray matter, if you don't remember, is the part of the spinal cord in this case that consists of actual cell bodies, soma, so your neural cell bodies. And the outer layer, which is termed the white matter, is what consists of myelinated axons. And it's called white matter because myelination causes axons to appear white. Now, in the gray region, the gray matter, we have three distinct regions we need to remember. First, the posterior gray horn, followed by the lateral gray horn, which is lateral, easy to remember, and the anterior gray horn, which is most anterior. So posterior gray horn, which is more posterior, anterior gray horn, which is more anterior, and of course the lateral gray horn. Then, surrounding that, of course, we have white matter, and just like in the brain where we have sulci, we also have sulci in the spinal cord, and we'll go over more of them as we progress through the lecture. But just for memory's sake, we have the first sulcus that I'm going to introduce to you. It's called the posterior median sulcus. So another thing to keep in mind is the presence of these nerve fibers. So we have dorsal nerve fibers and ventral. So dorsal nerve fibers are known as dorsal rootlets. So these are the ones that are carrying nerve fibers to the spinal cord. And the ventral ones are known as ventral rootlets. They eventually go on to form what are known as the dorsal roots and the ventral roots. And in particular for the dorsal root, there's a bundle of cell bodies which are called the dorsal root ganglia. And they contain the cell bodies of much of the sensory fibers that are coming from the periphery to your spinal cord. While the ventral fibers, they carry much of the motor information going from the spinal cord out to the periphery. And of course, these converge and form the spinal nerves, the bulk nerves exiting the spinal cord. So paying attention to the spinal cord as a whole, what you can see is that there are actually segments of the spinal cord that are much larger than the next. So we have two distinct regions, the cervical enlargement and the lumbar enlargement. So why these two regions are larger than the thoracic region, for example, or the coccygeal, That's mainly because we have a lot of sensory fibers and motor fibers that need to innervate the arms and the legs. So a lot of moving your arms and leg, locomotion, it takes a lot of nerve fibers to accomplish that, which is why you're going to be seeing this enlargement in those two cervical and lumbar regions. So cervical taking care of upper body, limbs, motor and sensory, and you're going to see the lumbar enlargement taking care of most of the lower limbs. Now, next on the slide, you're going to see different cross sections of the spinal cords themselves. So beginning with the cervical region, you can see that it takes this oblong shape. And you're going to see a higher density of white matter relative to the gray matter that's available. And if you're looking at the slides and you're questioning, hey, why is the white matter actually dark here? That's because of the staining that's done. So don't let that confuse you. That's very important. So as you can see, as you go down the spinal cord, so we're exiting the cervical region, it loses that oblong shape and eventually gains this more little circular shape. And the gray matter actually begins to thin out. So if you remember me mentioning the enlargements, a whole reason why there's this, if you take a look at the cervical sections compared to the thoracic, you can see that the gray matter, especially in the more anterior regions, are just more bulky, more full. That's because those are all the motor neurons that are taking care of your arm movements, whereas those are absent in the thoracic region. So hopefully that helps you understand the enlargements more. You can see that enlargement actually picks up if you get to the lumbar region, which again begins to round out, and actually you're going to see a higher density of gray matter relative to the amount of white matter that's present. So that's one distinctive point about the lumbar section. So next, we're going to actually take a look at the spinal cord model present here itself. So to begin with, I'm going to introduce to you the conus medullaris. Conus medullaris is actually where the spinal cord ends. And it's called conus because you can remember conus from the fact that it actually looks like a cone where the spinal cord comes to an end. Beyond that, we have the filum terminale. So terminale, you can remember a terminal or an ending, and that's actually the pia matter that attaches the spinal cord to the most coccygeal vertebra of the spinal cord itself. So it's a sort of latch for the spinal cord. After that, we have what's known as the cauda equina. So that's just the bundle of nerve fibers exiting the most sacral or coccygeal region of the spinal cord and exiting out to the periphery. So it's just these bundle of nerve fibers. And finally, we have the lumbar cistern. So the lumbar cistern is just, it's going to be difficult to visualize on this model itself, but if you can imagine I mentioned the pia and arachnoid matter earlier. And between them is the subarachnoid space, and that's filled with CSF, cerebrospinal fluid. And that extends down, and it passes actually the conus medullaris where the spinal cord ends and extends downward. So there's this region here full of CSF, and that's actually where the lumbar punctures take place, is in this lumbar cistern. So cistern is just a space filled with CSF. Just as a review, we're on slide 22, if you're following along. We're going to be looking at the different terms highlighted in red, and these you need to put into memory. So we have the lumbar and cervical enlargements. We have the nerve rootlets. I can show them here, that actually exit the spinal cord or enter the spinal cord, depending on if they're ventral or dorsal. We have the nerve roots themselves, so these rootlets eventually turn into roots, the dorsal roots and the ventral roots. Then we have the spinal nerves actually exiting the spinal cord. Next, we have this cauda equina, and that's going to be, if you remember from the slide, your horse's tail, right? These bundle of nerves exiting the sacral and coccygeal region of the spinal cord. The final filum terminale, which actually latches the spinal cord down to the vertebra, which is that pia matter. We have the cona medullaris, which is that region where the spinal cord actually ends. And of course, we have the vertebra, the bone itself. And one new term, it's actually going to be the sympathetic chain ganglia. And in this model here, you can actually find it here. And what it contains is the cell bodies of many of the nerve fibers important for sympathetic nervous system activation. So sympathetic chain ganglia. If you remember, ganglia is just a bundle of cell bodies. So talking about the autonomic nervous system now, autonomic nervous system itself is separated into two components. We have the sympathetic and the parasympathetic component. So the sympathetic is more of the fight or flight response, right? So what gets activated during stressful situations. And parasympathetic is what's active normally for you to be able to, for your cardiac system to function at a normal level, for you to be able to breathe, right? The control of breath that goes on in normal situations. So that's also referred to as the rest and digest portion of the nervous system. Now, what's important to keep in mind, there's a distinction between exactly where these systems, where the nerve fibers for these systems are actually localized. So for the parasympathetic nervous system, remember that's the rest and digest, normal situation, nervous system activation. The cell bodies are actually located in the brainstem and as well as the sacral region of the spinal cord. While for the fight or flight or the sympathetic activation, a lot of cell bodies are actually located in the thoracic and the lumbar region of the spinal cord itself. So again, the sympathetic chain ganglia is what contains another host of cell bodies important for sympathetic nervous system activation to even occur. So as I mentioned, there's thoracic and lumbar regions which contain cell bodies which then eventually send axons to the sympathetic chain ganglia, and they send it out to the rest of the organs in the body. That's how nervous system activation for a sympathetic response takes place. That's different from the parasympathetic. How? For the parasympathetic nervous system, you have your set of cell bodies in the brainstem or the sacral region of the spinal cord. They send axons out to these different clusters of neurons that are located actually very close to your organs. So there is no chain ganglia near the spinal cord like there is for the sympathetic nervous system. For a parasympathetic nervous system response, they send axons out to these peripheral clusters of neurons that are located near or in organs themselves. Now, going back, we're going to be taking a closer look at the model itself and going through the different sulci and just regions in general. So again, we have what is known as the gray matter and the white matter. Beyond this, I mentioned we have sulci. So the first sulcus I pointed you to was the posterior median sulcus. Now, if we begin medial and we go a little lateral on either side actually, we'll eventually reach what is known as the posterior intermediate sulcus. And the term will make sense if we continue a little more lateral and we reach what's known as the posterolateral sulcus. So how you can remember this is actually pretty straightforward. Posterior median sulcus, right, in the posterior region, and the median. And this is the posterior lateral sulcus, right? And between them is the intermediate. That's just an easy way for you to remember. Now, beyond that, we also have this central canal, which is actually where CSF resides, and it's an extension of the ventricles from the brain. Beyond that, we have, again, the separations that are outlined in the slide of the gray matter. So we have the posterior horn, the lateral horn, and as well as the anterior horn, right, or the ventral horn. So there are two ways to refer to these horns. You can say posterior horn or dorsal horn, anterior horn or ventral horn, as well as lateral horn. Now, in the white matter, there's also these distinct points or columns. Remember, white matter tracts, what they're important for is taking information from these cell bodies and taking them to the brain or to other regions of the spinal cord. So they're actually traveling a lot of up and down parallel fibers. So that's why they're referred to as columns. As you can imagine, if this was flat, they'd be columns sticking out, right, going up to the brain, down to other regions of the spinal cord. So in the most dorsal section, we have the dorsal column. In the lateral section, we have the lateral column, and the ventral column. So for the last part of lecture 3, we're going to be doing a couple of activities, one being 2-point discrimination, and the second one being the monosynaptic reflex, that is the knee-jerk reflex or the patellotendon reflex. So we're going to be going over these two concepts, and we're going to be doing some demonstrations that sort of give you a better idea of how they work. And you can follow along with at home with a partner. Beginning with 2-point discrimination. 2-point discrimination is a concept used in clinic to test whether or not the dorsal column medium lemniscus tract is actually present within patients. So whether it's functional rather than present. So with your partner, what you're going to do is you're going to ask your partner to please close their eyes and hold out their palm. So when they do that, you're going to take your calipers or your very sharp pencils or your paper clips, whatever you're using, 2 points. And what you're going to do is hold them out to a set distance apart from each other. And you're going to poke your partner's fingertips and ask exactly how many points they feel. So how many do you feel?
2.
All right, so she felt 2 points. So I'm going to take the same distance, and what I'm actually going to do is I'm going to poke her on another part of her body that's not her fingertips, that's not very sensitive, like her shoulder. So I'm going to poke her now and ask how many she feels. How many do you feel?
1.
So she felt about 1 there. So at the same distance, her fingers were more sensitive. They were able to feel the 2 points versus somewhere like her shoulder, or you can try it on your partner's back. That's the idea behind 2-point discrimination, is your fingertips, places that are important for fine touch, are more represented in cortex versus other regions, right, that you don't necessarily use all that often. So another demonstration we're going to show you now is actually another test that's done in clinic to test the response of a reflex arc present within the spinal cord. So this is testing the patellotendon reflex. And how it works is, again, you can try this with a partner at home if you have a tool as this one. What you do is you ask your partner to please close their eyes again. And what you're going to do is you're going to take this and gently bang it on your partner's patellar tendon, as so. So as you saw, her leg kicked out. So why you just saw that occur is because when I take this and knock it on her tendon, what I'm doing is I'm eliciting a stretch in her quadricep. It's a micro stretch. But what's happening is sensory fibers within that quadricep are actually being activated and sending these sensory relays to the spinal cord. Now, there are 2 things happen. One, it synapses onto directly a motor neuron, which is relayed back to her quadricep and causes that muscle to actually contract. And 2, it actually sends a relay to an interneuron within the spinal cord. And you can follow this along in your slides. That interneuron actually sends an inhibitory signal to the motor neuron, activating her hamstring muscles. So it's causing it to relax. It's not contracting. That's why it's easy for her to actually just kick out her leg, right? This whole motion is contingent on the fact that her quadricep contracts and her hamstrings remain relaxed.